realize like most of these words probably don't make sense right now, but I will explain. Okay, so my testable question is whether or not Chloroxys ostracis, which is a fungal species, can biodegrade high concentrations of polychlorinated biphenyl majorities toxins around like 500 parts per million in a controlled contaminated sawdust medium. So, and whether or not that degradation can be over 50%. And I have the same question phrase for another fungal species, trees versus corn. Exact same question. So, background information. Polychlorinated biphenyls are also known as PCBs. They are anthropogenic toxins made in the 1900s for industrial purposes. They have really high chemical stability, so they're used for like infusion and like a lot. They have this. They can be used a lot for many different functions. They are banned internationally when their toxicity was discovered, especially their environmental toxicity. They are very toxic because they're just very carcinogenic, and you don't want those substances like around you or inside of you. They tend to bioaccumulate, which means they don't like eradicate or like they don't disappear. They only build up in our environment. Like that picture over there, in water, the concentration of like PCBs is very small, but like they, since they bioaccumulate, they build up within the tertiary systems. Like they become higher as you go up the kingdoms. Like birds of prey, they have the highest compared to like the water. So yeah, most destruction methods fail. So many scientists are looking forward or like looking into like better ways of remediation. White rock fungi, they are a type of fungus that have lignin degrading properties, meaning they have these enzymes that work really well in degrading many substances, which is why I thought, hey, like why don't we, since they've been used to degrade other substances like hydrocarbons and pesticides, I thought, hey, why not try these on PCB, which are another persistent compound. So they're very good in breaking down the chemicals into like less harmful substances. The GCFID is a piece of machinery that I will be using for like analyzing the total content of the toxins of polychlorinated biphenyls. They're just gas chromatography is the official term for it. It's just to refer to this like analytical technique to separate compounds and gas. The pictures over there, they're just like how the machine works. So I'll be injecting my sample through the injector port, like going through the column of it. The detector, that picture on the right, is like a close-up of the detector. So essentially, my sample's going through the column, and helium and like these other like substances, they are also going into the column, and essentially my sample is being burned at very high temperatures, and the cathode at the top is collecting the ions released from this thermal decomposition of my sample, and it's basically just recording the components of the sample through like the analysis of the ions. I could get further into it, but like I'm still figuring out how it works. It's so complicated. Okay, so my hypothesis is I believe that if I add pre-cultivated Chloroxys ostracis to PCB contaminants, that the mycelium, which is just like a component of the fungi, then I believe it will degrade the PCBs due to the enzymes in the fungi mycelium. And same with Trinides versus color, which is just another fungal species that is white rot, so it has the same like degrading, high biodegrading tendencies. I measured my results through the gas chromatograph and the CEM solvent extractor, which is that I'm doing at a different lab. Independent variable is the use of different fungal strains. So I'm using processed osteosis, which is oyster mushrooms, and I'm also using Mount Tinua. I'm using Tuberous versicolor, which is like another type of mushroom. Dependent is a total PCB quantities, so that's being measured through the gas from abstract, and obviously controlled is just like sawdust contaminated mixture that does not have any fungi added to it. Here's my materials. I'll explain them further in my procedure. So part one of my procedure is basically I am getting these glass containers in which I'm putting my contaminated sawdust sample, where it's contaminated with PCBs, and I'm adding them to the glass containers. And later on, then I will place my control away. Then I'm going to add fungi to inoculated fungi, which is just basically sawdust that has fungal strains within them, and I'm going to add them to the sawdust samples that are contaminated. And basically, I'm going to incubate them and allow the mycelium, which is a part of the fungi, to spread throughout the entire sample. And once that's done, I will take them to the lab to extract after 2 weeks that they've been incubating. I'll take them to the lab, and I'll prepare these reagents and do this sample extraction at the lab with this CEM equipment that I'm using. Basically, I have to make a bunch of hexane and acetone reagents and add them in various mixtures and put them into this machine. Basically, I bring a solid thing into a machine, and it turns out into a liquid thing that I can put into